Alright, so next we're going to do the isolation procedure to isolate the EGFP plasmid from our bacteria cultures. So you're going to take 1 milliliter of bacterial culture and add it to a 1.5 milliliter Eppendorf tube. And then you're going to centrifuge it at 6,000 RPM for 2 minutes. You're going to use the centrifuge machine, so you hit this button here with the lid and the arrow to pop the lid up. Then you want to push in the red part and lift up to take the lid off. Then you're going to put in your sample, and you always want to be sure to balance your sample in the centrifuge. So place another tube of equal weight right across from the tube you have originally. When you guys are going to be doing this procedure, you're going to have 2 tubes that you're going to be doing because you'll be doing 2 plasmid samples, so you can just use that to balance. Then when you put the lid back on, you want to push it down until you hear a click, like that, and then put the lid down, and then you can set the RPM to 6, and we're going to run it for 2 minutes, and then you can just hit the play button and let it run. So once it is done with the 2 minutes, you can let the RPM go down to 0, and it'll beep to let you know. Then you just hit the button again to take the lid off and push in the red lever to take the lid off. Then you're going to take out your sample. As you can see, there's the bacteria pellet at the bottom and then a kind of yellowish supernatant on top. So what we're going to do now is we're going to pipette off that yellowish supernatant without disturbing the bacteria pellet. And you can just dispose of all that media in a beaker. Okay. And then what you should have left is just the pellet with a little bit of media. Alright, and then you're going to add 200 microliters of your resuspension buffer 1, which is the pink one. So then just take up 200 microliters and add it into the sample, and then you're just going to pipette up and down to break up the clump. So just take the liquid up into your pipette and pipette it back down a few times until that clump is completely broken up. And it should look like this. Then you're going to add 200 microliters of the lysing buffer 2. Going to invert it just by tilting it upside down like that, like 6 to 8 times. It should get that clear kind of pinkish color. After that, you're going to add 400 microliters of the neutralizing buffer 3. And once that's added, the solution should turn yellowish, and you should see a white precipitate starting to form. And add the 400 microliters. Right. again, we're going to mix it by inverting it a few times like that. Again, you don't want to shake it, you just kind of want to invert it. And as you can see, it's turned yellowish and you can kind of see that white precipitate. So then after this, we're going to centrifuge it for 3 minutes at 6,000 RPM. So again, you want to put it in and make sure you're balancing it with another tube of equal weight. And then push the lid down until you hear that click. Then put down the lid. And then you want to set it to 6,000 RPM for 3 minutes. And then you can go ahead and hit play and let it run. And if you don't balance the centrifuge correctly, you'll hear that it isn't balanced correctly, and you'll be able to hear it going around, which means you should stop it and make sure that you balanced it. Alright, so then just go ahead and let that run for 3 minutes. Alright, so then once it's done, you can go ahead and pop the lid and take out your sample. You can see the clear, like yellowish supernatant and a kind of bluish white precipitate at the bottom there. Next, we're going to pipette 600 microliters out, and then we're going to put that into the top of the spin column. So we want to be careful to get up 600 microliters without disturbing the pellet that's at the bottom. Slowly and carefully take out 600 microliters. And then you want to just put that in the top of the spin column like that. Alright, and then next you're going to put the whole spin column into the centrifuge and centrifuge it for 60 seconds at 6,000 RPM. And it doesn't have a lid on it, but that's okay, it doesn't need one. It's not going to flow out over. So just put it in and then make sure that it's balanced again. And then put the lid on. And then we're going to let it run for 6,000 RPM for 1 minute. That's done, you can go ahead and take out the tube. And you can see that a lot of the supernatant has kind of flowed through to the bottom. So we're going to take out the top column and then just jump out the flow through. Then we're going to put the spin column back in. And then we're going to add 200 microliters of the wash buffer 1 back into the top of that spin column. So 200 microliters of wash buffer 1. And then just insert that into the top of the spin column. And then we're going to centrifuge it again for 1 minute. And again, you don't need a lid or a cap for the sample. Once again, you can see the flow through has gone to the bottom, so we're just going to take out the top and dump the flow through out again. And then this time we're going to add 400 microliters of wash buffer 2. And add that to the top of the column. And then we're going to centrifuge it again for a minute. Take out our sample again. We're going to discard the flow through again. And then this time we're going to take the spin column and put it into a new clean centrifuge tube. And then we're going to use a P200 to take up 50 microliters of DNA elution buffer. And then we're going to put that into the top of the spin column. And then we're going to put that in, and then you want to balance it again, but you can just use an empty 1.5 milliliter centrifuge tube, right? And then you want to close it again, and we're going to let it stand for a minute before we centrifuge it again at 6,000 RPM for another minute. So you should just let it sit for a minute. You can go ahead and centrifuge it at 6,000 for a minute again. Okay. So you can take it out. And then as you can see at the bottom there, that clear liquid is the 50 microliters of buffer that has your eluted plasmid DNA in it. So you just want to take out the top of the spin column, and then make sure that you label the tube with all of the necessary information, and then you can give it to a TA or instructor to store. So spectrophotometers are used to qualify and assess the purity of a molecule based on its absorbance at a specific UV wavelength. So we're going to be using the Nanodrop spectrophotometer to evaluate our DNA samples. Nanodrop spectrophotometers use 1 microliter instead of the 3 milliliters that the regular spectrophotometer that we used in Module 1 uses. So you're going to need to compare the results from the Nanodrop spectrophotometer to your agarose gel results to verify the yield and integrity of your sample. So remember that one strong band in your gel is good, while multiple smaller bands indicate like fragmented DNA. So once you're ready, you can go ahead and select double-stranded DNA on the Nanodrop. And you can notice too that the Nanodrop can also evaluate single-stranded DNA, RNA, and proteins. So we're going to select double-stranded DNA. And then you're going to take 1 microliter of water up with the P10, and then you're just going to put it on the base of this pedestal, trying to get it right on top of that black dot. And then you're going to take a Kim wipe and just wipe off the bottom and the top of the pedestal. Then take up another microliter of water using your P10 and put it on the black dot in that silver pedestal again. And you can use your other hand to steady the pipette if you need to. Then you're going to close the arm gently, and it should auto blank, but if it doesn't, then you can go ahead and hit blank. Okay, and then once it's blanked, you can lift back up the arm, and then you're going to take another chem wipe and wipe the blank off of the pedestals. Then you're going to take up 1 microliter of your DNA sample. And then put that onto the pedestal, right on top of the black dot. And then you're going to gently close the arm, and it should auto measure, but if not, again, you can hit measure. And then your results should come up. So there's a few different things that we're looking at with the results. So first of all, the height of the peak on the graph indicates the amount of DNA. So the higher the peak, the more DNA you have. And it's also indicated by the number here, which is in micrograms per milliliters of DNA, which indicates the concentration of DNA in your sample. So you can see this sample had 208.6 micrograms per milliliter, which is a pretty good amount of DNA. You need above 50 micrograms per milliliter for your DNA sample to be usable, and then anything from 300 or above would be indicated by a strong band on the gel. And then another thing that we can look at on this is the steepness of the peak. So as you can see, this peak is fairly steep, but it could be a lot steeper, indicating a cleaner sample. And then this is also indicated by the 260-280 ratio, which, like the steepness, indicates purity. So 260 nanometers is the wavelength that DNA absorbs at, and 280 nanometers is the wavelength that other macromolecules and contaminants absorb at. So having a 260-280 ratio of 1.8 to 2.0 indicates a good pure sample. So this sample has a 260-280 ratio of 1.85, which is good. So you can see that this is a relatively pure sample with a high concentration of DNA. So when you're done looking at all that, you're going to want to photograph that screen to have that to work on your worksheet and report. And then you can just hit end experiment and then hit end experiment again. Often research requires large amounts of DNA or a specific segment of DNA for analysis. In this activity, we will use an enzymatic repetitive amplification to generate an increased amount of the target fragment or segment of DNA that you want. The technique to do this is called polymerase chain reaction, or PCR. In this activity, you're going to amplify the green fluorescent protein, or GFP. GFP is used as a tag to monitor the location and level of protein expression in cells. The protocol uses a DNA polymerase that is heat stable and is called Taq polymerase. It was first isolated from the thermophilic bacteria Thermus aquaticus. The basic steps require mixing a trace amount of GFP template DNA with short DNA primers for the 5' and 3' ends of the GFP DNA and the Taq enzyme. The reaction along with the control reactions are set up in an 8-reaction PCR strip tube. The green color is going to be used to monitor the DNA migration in the agarose gel, and this is the 2X Taq PCR master. So the amplification is going to be automatically repeated for 30 cycles, and each cycle consists of 3 different steps. So the first step is the heat denaturation to separate the GFP DNA into strands. Annealing is the next step, which is a cooling step to allow the primers to anneal to one strand of GFP. And the final step is extension, where the Taq polymerase uses the primer and template to extend a new strand of complex DNA. So the thermocycler is already set up. So first, you want to open the lid and put your 8-strip tube inside, and then close and lock the lid. Then you're going to hit Setup Run, and then you're going to hit Open Method, and then select GFP 2016, and this will display a diagram of the steps and the temperature at which they run. So the 95 degrees Celsius step is the heat denaturation to separate the DNA strands. The 60-degree step is the annealing, which cools and allows the primers to anneal to one strand of GFP. And then the 72 degrees Celsius step is the extension, where the Taq polymerase uses the primer and template to extend a new strand of DNA. At the end of the 30 cycles, an instructor will recover to the freezer your sample